In this video, we're going to talk about the transcriptional regulation that occurs within prokaryotes, within these single-celled organisms. Now, prokaryotes, bacteria, and archaea are, as I said, single-celled. And so what that means is all the genes contained within that cell are obviously necessary for that cell to survive. And so why is it that there's regulation, right? Why doesn't this single-celled organism make all of its genes or turn on all of its genes all of the time? Well, the answer is that even though all of these genes are necessary for the survival of these cells, it doesn't necessarily mean that all of the genes are necessary all of the time. And so if there has been a regulatory system evolved that gives the cells the ability to turn off the activation of genes that aren't necessary at a given moment in time, those cells will have an evolutionary advantage over cells that have not evolved a system of regulation. And so therefore, over the course of millions and millions of years, then, these systems of regulation have evolved. And so we're going to take a look at that. And first we're going to look at, in general, what is necessary for transcriptional regulation. And then I'm going to have you watch a few videos that about various regulation systems. Okay, so here we have gene expression. Now, gene expression includes the transcription of the gene within DNA into messenger RNA, and then the translation of that messenger RNA into protein. Okay, and then that protein is modified in various ways so that it's finally functional, and then that has a role in giving rise to a particular trait. Maybe the trait is to be able to metabolize lactose. Maybe the trait is to create tryptophan. Okay, now, there are many levels at which this process of gene expression can and are regulated. The most common level at which gene expression is regulated is right here, at whether or not a gene is going to be transcribed in the first place. Now, there are several things, regardless of whether we're talking about transcriptional regulation in prokaryotes or eukaryotes, there is some commonalities, some absolutely fundamental commonalities. And those are that we need a sequence of DNA called regulatory sequences that are going to determine whether or not a particular gene is going to be expressed. And we need the proteins that bind to those regulatory sequences called specific transcription factors, or sometimes activators and repressors. So here are the regulatory sequences. And you can see that regulatory sequences are necessary no matter what type of cell we're looking at, whether it's a bacterial cell, yeast cell, or human cell, within the DNA, within the gene, within those cells. Now, the difference is that in bacteria, those regulatory sequences are often very, very close to the promoter. And that'll become important, and we'll talk about exactly why that's important a little bit later. Then when you start to get more and more complex organisms, especially multicellular organisms, you get a situation in which these regulatory sequences are still associated with a single gene, but they can be very, very, very far away from that gene. They can be upstream of that gene. They can be downstream of that gene. Right? So it gets to be pretty complex. Now, these sequences of DNA that I'm giving the generic term regulatory sequences, have lots and lots of different names. They can be called enhancers and control elements and proximal control elements and distal control elements and activator binding sites. So make sure that you are aware that all of these terms are examples of regulatory sequences. Sequences that are associated with the gene that determine whether or not that gene is going to be activated, whether it's going to lead to the transcription of the gene's messenger RNA. Now, The importance of these sequences are that they are bound by specific transcription factors. Now make sure you understand, I'm using that word specific specifically, because it's to distinguish these from general transcription factors. Those are the transcription factors that are associated with RNA polymerase at every promoter. Your TF2D, your TF2A and B, those are general transcription factors. These are specific transcription factors. They do not bind to promoters. They instead bind to regulatory sequences, okay, to these various enhancers, control elements, etc. They do not bind to the promoter. Now, what do they do? Well, this is what they do. They have a domain, a portion of the protein that is called the DNA binding domain, and that domain of the protein gives them the ability to bind to very specific sequences, okay, called their DNA binding site. And then there's another portion of the protein called the activation domain. And this is the part of the protein that then binds something else, usually another protein. Now, if we're talking about a specific transcription factor in prokaryotes, in bacteria, oftentimes the activation domain binds to RNA polymerase itself. It acts like another contact for that RNA polymerase to the DNA upstream of the gene. In eukaryotes, it's rarely the case that the activation domain of a specific transcription factor interacts directly with the RNA polymerase. More often than not, it recruits maybe the general transcription machinery like the TF2A and B and so on and so forth, or chromatin modifying factors. So we're going to look more at that when we talk specifically about the regulation of transcription in eukaryotes. Okay, now these specific transcription factors, also called activators and repressors, are special in that they have DNA binding domains that bind to what? That bind to regulatory sequences. And that's it, okay? But as simple as that sounds, this whole idea of transcriptional regulation can get really confusing really fast. But that will become more important to understand when we get to eukaryotic transcriptional regulation. All right, now if we look at a whole bunch of specific transcription factors and we compare all of their DNA binding domains, we find that there are various families. Okay, there's more than what I'm just going to show here, but I want to give you an idea of some. All of them oftentimes bind as homodimers and bind therefore to these half sites. So for example, the lac repressor that you're going to look at very soon binds as homodimers, and so you have these two little half sites that they're going to bind to. And they do so by interacting with the major groove. So if we look at something called a helix-turn-helix domain, meaning helix-turn-helix domain, we can see that one of the helices is what's called a recognition helix. So this alpha helix 3 here has amino acids that can hydrogen bond very specifically with elements in the major groove, and that's the way it reads the sequence of DNA without separating the two strands. Okay, we talked about the major groove like the first lecture of this course. Here we have that information that we discussed on display. There is another DNA binding domain called a zinc-containing domain. And so zinc acts as a cofactor here in the protein. It stabilizes the structure. But we still have a sort of recognition helix. There it is, this alpha helix that's going to bind to the major groove of the DNA, and it's going to interact very specifically with specific hydrogen bonding interactions and bind to very specific sequences. There's a homeodomain. Okay, this is again, a homeodomain is another DNA binding domain. It's encoded by the homeobox portion of a gene. And it has that same idea of a recognition helix. Now, what separates it from the other two is how that recognition helix is stabilized. But we have, again, a recognition helix that has various R groups of amino acids that interact with the edge of the major groove, with the various bases in the major groove. Now, HOXB1, the gene that you guys are reading about within the paper, encodes a protein with a homeodomain. Now, of course, that paper is only discussing the regulation of the transcription of that gene and not the protein, but I just want you to be aware that the protein encoded by HOXB1 is a homeodomain protein.